Hello, everyone. Now we are going to have a session on chapter 5th, which is on translation. And this chapter 4th and 5th, they come together. And I have shared with you on the blackboard as one complete chapter, as one. So I covered in the last recording transcription. Now I'm beginning with the translation, okay? So let's go to the slides, and I would like to start here. So if you notice, I have paid a lot of importance to this figure to highlight the complexity in eukaryotic transcription, post-transcriptional changes, which are capping, poly-A addition on the 3' end, splicing, then export to the cytoplasm for translation, which is very much contrast to bacteria or prokaryotes, where it's almost a straight line. There is no post-transcriptional events. So what are the post-transcriptional events? Capping, polyadenylation, and splicing. So these are, and then the mRNA goes to the cytoplasm, to the ribosome, and essentially for translation, right? Now, in bacteria, there is a special sequence called as Shine-Delgarno sequence. This sequence helps the bacteria to help the mRNA of the bacteria to recognize the RNA on the ribosome. So why this? Because when mRNA is traveling to ribosome, there has to be a launching pad where the mRNA has to bind to the ribosome. And in bacteria, this sequence is called as Shine-Delgarno sequence, right? It's a ribosomal binding site for mRNA, right? Now, let's review before we enter into the details of translation. What are the salient differences between prokaryotic and eukaryotic transcription? So as I mentioned to you, in prokaryote, it is continuous, whereas in eukaryotes, there are post-transcriptional changes, then export to the cytoplasm through nuclear pore. And in prokaryotes, the 5' end of mRNA is immediately available, meaning that there is no capping in the prokaryotic mRNA as compared to eukaryotic mRNA, which has to go capping and polyadenylation plus splicing. Pay attention to the ribosome type. The ribosome size or density is 70S in prokaryotes, whereas ribosomes in eukaryotes are bulkier, 80S. Also, ribosomes have two subunits, one larger and one smaller, which are 50 and 30 in bacterial ribosomes, and which are 60 and 40 in eukaryotic ribosomes. And also pay attention to the larger subunit of ribosomes with two rRNAs, molecules 5S and 23S, whereas larger has three rRNAs, 5S, 5.8S, 28S. Three different kinds of ribosomal RNA in the larger subunit of ribosome. Smaller subunit has 16S, whereas humans or eukaryote have 18S. Smaller subunit of ribosomes have 21 different proteins, whereas in eukaryote, smaller subunit of ribosome have 33 proteins. Similarly, there are 36 proteins in larger ribosomes and 49 proteins in larger ribosomes of eukaryote. So I almost read all the differences to you. Now let's come to the essential players of translation. So the essential players are are mRNA, tRNA, and rRNA, plus ribosomes, and there are many factors that are involved, right? But essentially, this is a well-managed orchestration of great work on the surface of ribosome, right? So in eukaryotes, a primary transcript is formed, which is nascent, which undergoes capping, poly-A tail, then splicing. After this, the mRNA becomes mature and it is ready to go to the cytoplasm. And in the cytoplasm, it wants to meet the ribosome, right? So this is what happens. The mRNA travels through the cytoplasm, goes and binds to the ribosome. And the purpose of binding to ribosome is that the tRNA can come with specific amino acid. They come at the surface of the ribosome. And on the surface of the ribosome, there is a peptide bond formation, and there is a growing chain of polypeptide, right? So single amino acids are brought to the mRNA at the surface of ribosomes by the tRNA. On the surface of ribosome, the tRNA and mRNA, they talk to each other, they bind to each other, and then there is a covalent bond formation on the larger subunit of the ribosomes, right? So let's go more deep. So what you see here, that a ribosome has a smaller subunit and a ribosome has a larger subunit. On the smaller subunit, there is a recognition of mRNA and tRNA take place on the smaller. On the larger subunit, peptide bond formation takes place. So tRNA comes and binds to the mRNA, and the binding is through hydrogen bond that we will see later. And the site at which the tRNA comes is A site, and the site at which the growing chain of peptide is growing is P site. But we will go into more details in upcoming slide. Now the biggest question that should come to you, or you should, so mRNA, what is mRNA? mRNA is a stretch of ribonucleotides. On this mRNA, 3 nucleotides are called as triplet nucleotides. They come together to code for one amino acid. So these triplet nucleotide is also called as codon, right? So this AUG, it becomes triplet codon, and then it codes for amino acid methionine. Then GUC codes for valine, AGU for serine, CCA for proline, and there is a stop codon. So very briefly, how this relationship happened. So this relationship between mRNA and respective amino acid through triplet bases happened by the work of Hargobind Khorana, who solved this mystery that triple nucleotides or ribonucleotides on the mRNA codes for one amino acid, right? And he got the Nobel Prize, and that is called as genetic code, right? So there are, we know that there are 20 amino acids, right? That was the biggest puzzle that came to every scientist or molecular biologist, to draw a relationship between mRNA and protein. Why it was a puzzle? Because there are 20 different kinds of amino acid, but essentially there are only 4 nucleotides in the DNA that would transcribe into 4 nucleotides in the mRNA or RNA. So there is a direct relationship between RNA and DNA when RNA is synthesized, but there is no direct connection between mRNA and protein because there are 4 bases and there are 20 amino acids. So they did a lot of experiments by doing synthetic chemistry, synthesizing artificially mRNA, and they came to a conclusion that 3 nucleotides on the mRNA, it serves to code for an amino acid. And this they presented as a table called as genetic code. So pay attention. So in this side, on the left hand column, there are 4 bases, U, C, A, G. Why U? Because this has to do with mRNA, RNA. And also here, 3 bases. And here, 4 bases and 4 bases. These give rise to 4 times 4 times 4, 64 triple-nucleotide situations, right? Out of this 64, 1 is start codon, AUG, 3 are stop codon, UAA, UAG, UGA. So out of the 64, 3 are stop codon, and rest 61, they code for amino acids. Now you would notice here that it would raise another question from other end, that why do we have 20 amino acids, but 61 different triple nucleotides situations, right? So that is to give flexibility to the system. For example, if you look here, CCU, CCU, CCC, CCA, CCG, all these 4 codes for amino acid proline. Again, 4 codes, 4 triple nucleotides or 4 codons for leucine, right? So one amino acid could be coded by multiple triple nucleotides or multiple codons, and that perhaps makes the translation an efficient process, right? So this is the story about mRNA. That mRNA stands for messenger RNA. And what is the message? The message here is to build a relationship between triple nucleotide and an amino acid. But where does this amino acid comes? This amino acid is brought to mRNA by another RNA called as tRNA. And where this is happening at the surface of ribosomes. And to be more specific, tRNA and mRNA, their recognition happens at the smaller subunit of the ribosome. So let's look at the tRNA. So tRNA has a very unique structure, right? They are like a clover leaf. So what you will see here, 3' end and a 5' end, right? And then you will see some bulging loops, right? Bulging loops. They are formed, these loops are formed because they are interspersed or placed between double-stranded RNA. So tRNA is a mix of double-stranded RNA or single-stranded RNA. At some place, this single-stranded RNA, which cannot form double-stranded, forms a loop. And one such loop is called as anti-codon loop. This anti-codon loop is the loop that binds to the mRNA. Remember, I mentioned to you that the recognition between tRNA and mRNA occurs on the smaller ribosomes. Ribosome has 2 subunits, large subunit and smaller subunit. So this codon and anticodon. So anticodon from tRNA and codon from mRNA. They talk to each other at the lower surface of the lower subunit of the ribosome. Now, the question is, how come tRNA gets the specific amino acid? So let's consider an example. So the anticodon here is ACC. So anticodon will bind to a codon on mRNA that will be UGG, right? So this UGG codes for amino acid tryptophan. So tryptophan amino acid will be brought by specific tRNA, which has an anticodon of ACC in the anticodon loop. Now, how this amino acid will lodge onto tRNA? So this is a energy-based reaction. So ATP is hydrolyzed into AMP. Most of the time, ATP is hydrolyzed to ADP and single inorganic phosphate, but here it is pyrophosphate, AMP. So remember this, right? So the covalent linkage of amino acid and tRNA occurs, right? And this is catalyzed by an enzyme called as tRNA synthetase. In here, it is amino acid is tryptophan, so it is called as tryptophenyl tRNA synthetase. So the tRNA is charged with the specific amino acid, right? And here, the base pairing takes place between the codon of mRNA and the anti-codon of tRNA. So let's now go to another member called as ribosome. So ribosome, as I mentioned to you, is made up of two subunits, large subunit and a small subunit. And you can see this large and small is because of the density, right? Molecular weight, right? And they are made up of these ribosomes. They're made up of specific proteins and a specific RNA, right? But they have very important role. Now I want you to focus on here again, same message, but see here, prokaryotic RNA or ribosome is 70S. The large subunit is 50S and smaller subunit is 30S. Similarly, eukaryotes 60 and 40S and they make 80S, right? And this is the sequence of mRNA to which amino acid is brought by tRNA, right? And then the ribosomes slide across the mRNA and they keep on encountering new triplet nucleotide, new amino acids. They come until and unless the ribosome encounters the stop codon, right? UGG, UGA, and UAG. So ribosome here is a very complex structure, right? It's a dense structure of proteins and rRNA. Right? So many secondary structures, right? But they help in the formation of peptide bond. So let's see. So I mentioned to you that mRNA moves out of, in case of eukaryotes, it moves out of nucleus as a mature RNA and goes to the ribosome. It binds to the smaller subunit of ribosome, and then tRNA, it comes with specific amino acid, right? And then the peptide chain is growing. And then the ribosome slides to the new base, then new base, and each new codon rather than base, and each codon, which is triplet nucleotide, for every triplet nucleotide, there is a tRNA that comes with a unique amino acid, which is complementary to the codon and anticodon. So there's a very specific relationship between tRNA and mRNA. Now, as I mentioned to you, this process of translation is very planned, right? Planned in the sense that there is an initiation when the ribosomes and mRNA interact, and then there is elongation when the polypeptide chain is growing, right? A protein could be made up of 10 amino acid, 100 amino acids, or it could be 2000 amino acid. It depends, right? So the longer the mRNA, longer the protein, right? And then at some stage, the chain of protein doesn't grow forever. Every protein is made up of a specific number of amino acid. That's because there is a stop signal through stop codon, and the protein length is determined by that. So this example I already gave you, that codon and anticodon interaction is important for synthesis of polypeptide chain. Now, I'm going to a very important slide. This is the most important slide, which would help you understand the process of translation. Right? So ribosomes have three sites, right? An A site, a B site, and E site. A site is referred to as acceptor site, right? On this A site, always a new tRNA with amino acid comes, right? Then there is a P site, and then there is an E site. The P site is responsible for the growing polypeptide, right? And then E site is exit site. Now exit site for that tRNA which has dislodged its amino acid. So this is let's say tRNA that brought amino acid number 2. Now this tRNA dislodged its amino acid number 2 here, right? So now this tRNA is empty. It has to go away. So it goes from an exit site, right? So, and then when these 3 events happen, then the ribosome has to shift to the next triple nucleotide on the next codon. And again, a new tRNA will come. This acceptor site will become P site, and there will be a covalent bond formation for the growing polypeptide chain. So let's again revisit this, because this is every slide I'm trying to recapitulate the basic and try to advance also. So let's say this here, the ribosome has translocated, has slid across the triple nucleotide 1 codon, right? So this acceptor site is empty, right? On this P site, the 4th tRNA has come and it's sitting, and there is formation of peptide bond between amino acid number 3 and 4th. Now, since the peptide bond formation has taken place, the 3rd tRNA, there is nothing there, so it has to be exited out, right? So if you see here, the tRNA has exited out. Now on this acceptor site, the 5th amino acid comes, right? The 5th amino acid comes. Now what has to happen? That this ribosome has to go to the new triplet codon. When it goes to the new triplet codon, the 5th will form peptide bond with the 4th amino acid. And when the 4th amino acid peptide bond is formed, this site will become E site and it will be ejected off, right? So this is what in a way is happening. The next acceptor site was, unfortunately, the stop codon, right? And when stop codon comes in, the ribosome has to stop. The release factor has to come and bind to the stop site, right? What is the stop site? Stop codon. And beyond this, there won't be any protein synthesis, right? So here, the peptide chain or peptide bond formation takes place. There is nothing on the EE, and then the protein has to be released from the ribosome. So same slide here. The way termination is happening, the protein is released. Now this is also synopsis of initiation process. Stop. So there is, on the mRNA there's a start codon and there is a stop codon. So this is the start codon and a stop codon. And between this start and a stop codon, there will be nucleotides, and nucleotides in the partnership of 3 nucleotides, which work as a codon, and each codon codes for 1 amino acid, and that is present in the genetic code, right? So recognition happens here through codon and anticodon relationship. The first amino acid, this is very important. The first amino acid, it always comes on the P side, right? And rest, all the amino acids, they come on the A side. So this is the first amino acid, it comes on the P side. So let's say AUG means methionine. The next amino acid, it comes on A side. And then the ribosome has to shift to a new position, right? When it shifts to a new position, the A side becomes P side and the E side becomes, the P side becomes E side, and its ribosome slides. And the peptide bond formation takes place on the larger subunit of the ribosome, right? So this is also the same kind of explanation that happens in eukaryote. Now, this is the most important slide because ribosomes are involved, which is a very large particle, or one of the largest particles inside our cells, and it has to move from one codon to the next codon, right? So it needs energy, right? So the ribosome binds to the first codon, the first amino acid comes. Now the second amino acid comes at the A side, right? So this cross, then A side, right? Then a peptide bond formation takes place. Then the ribosome has to move. Now this is an energy needing process. What is the energy that comes here? It comes in the form of GTP, right? As compared to ATP, here the energy is GTP. So the new tRNA comes with EFTU factor, which has GTP. This brings the tRNA, and then this GTP is hydrolyzed into GDP. And once it is hydrolyzed, then it is again recycled into GTP, this energy process. Now during this energy process, peptide bond formation takes place. And again, the ribosome now has to move through 3 nucleotides. So again, another GTP is required, but another factor is responsible here, EFG. So first factor is EFTU GTP that comes with tRNA, and the second is EFG, which is a sort of an elongation factor that helps the ribosome to slide across the mRNA until it meets the stop codon. And again, EFG and EFTU, they're both GTP. The GTP is hydrolyzed to GDP, and this energy is required for the formation of peptide bond as well as sliding. So remember EFG versus EF2U. Last slide is that, which is very important from application point of view. We all consume antibiotics in case of bacterial infections. And these antibiotics, they target mainly the transcription and translational machinery. So tetracycline, it blocks amino acyl tRNA to A site of the ribosome. It blocks the binding of tRNA to the A site. So this tRNA cannot bind, due to which the bacteria cannot synthesize proteins, and bacteria will either stop growing or die. Similarly, streptomycin, it prevents transition from initiation complex to the ribosome elongating, so stops the initiation process. Chloramphenicol, it blocks the peptidyl transferase reaction on the ribosome. So if peptidyl reaction doesn't happen, then peptide bond is not formed. So the whole complex will fall apart. Cyclohexamide, it blocks the translocation reaction of ribosome. Rifampicin, it blocks the initiation of RNA chain. So this is the process of translation. So here we finish this lecture.